Hi, good morning, everyone. Welcome to the GenCefe webinar on safe protein synthesis technology and its applications in membrane protein production. I'm Serena Gom. I will be host today's meeting. Before we delve into our topic, I kindly to ask everyone to please mute your microphone, and thank you for the cooperation. Today, we will be very honored to have the Professor Lei Kai to give us a talk. So Professor Kai is the co-founder of the GenCefe and distinguished figure in the field of the safe protein synthesis. He previously worked at the Max Planck Institute of the Biochemical, where he leads the safe protein production group. Dr. Lei Kai specializes in the safe synthesis biological in the underscored by his extensive published record, with over the 40 papers in esteemed journals such as the ACS Synthetic Biological, New Biotechnology, and the JACS and Nano Letters. So without further ado, I invited Professor Kai to commence his presentation. Dr. Kai, you could share your screen so that we can see your screen.
Okay, can you hear me? So, can you see my PowerPoint presentations?
Yeah, we could see your screen.
Okay. Thanks for the host or the chairs give me the opportunity to share the exciting cell-free protein synthesis technology to colleagues and friends, in particular from industry. So today I'm going to focus on the application of cell-free techniques on the production of membrane proteins, which is a topic that I started back to my PhD times. And to my opinion, the cell-free protein synthesis techniques is especially suited for this type of difficult protein in terms of tools for recombinant protein production. Aligned with today's webinar talks, I divided today's talk into two major parts. First, I will give a brief introduction about cell-free protein synthesis. And second, I'll move on directly to membrane protein production using cell-free. And first, I'm going to give the general approaches that we applied in tackle the challenges for produce membrane protein recombinantly using cell-free protein synthesis systems. And the last, I will give some examples that we have investigated using cell-free systems. Okay, first, what is cell-free protein synthesis? In one word, is to move the central dogma from cellular environment into a test tube. That we reconstitute all the transcription and translation machineries in a test tube, which originated from cellular machinery. And this including transcription and translation pores of enzyme. And the transcription, which is carried on by RNA polymerase, and the translation was coordinated by the ribosomes. And of course, the translation factors and also amino acid tRNA synthesis and also other enzymes involved during the whole processes. And besides that, there is also a small part of substrate, which including amino acids and nucleotides, and also buffer salts and energy components, which sustain the whole reactions, which can continuously take place. And of course, the template of your gene of interest. Before I move to details, I'm going to give a brief description of the history that cell-free protein synthesis techniques has went through. So back to the late 80s, Edward Buscher already used the yeast extract to convert from sugar into alcohol. This already, I think, the prototype of what we mean today, the cell-free techniques. And a milestone achievement was done by Nuremberg and Mattei in 1960s, where they use cell-free translation system, which they isolate ribosome from the E. coli cells and perform the translation from polynucleotide template. And they finally, using this approach, decoded the genetic code. And later on, a set of individual work from Zubai, Alexander Spiering, and also James Watts and Shimizu or Endo Yuda from Japan, they all contributed the continuous development of cell-free techniques so that it's mature enough to become independent recombinant protein production techniques, which in parallel to what we use today frequently as eukaryotic or prokaryotic cellular expression systems. And this including continuously increasing the productivity of cell-free protein synthesis from several micrograms per milliliter up to today around several milligrams per milliliter. So this is really a dramatic shift of efficiencies of cell-free systems so that it can be productive as the cellular system, while still specialize in tackle the challenges from difficult proteins. While what we see today is very diverse cell-free system, so we can categorize, classify the cell-free system based on their cell lysate originate. For example, if the cell lysate is come from E. coli or can come from wheat germ or insect cells or the frequently used rabbit reticular oocytes, and also today there are several mammalian cell lines like CHO or HeLa, and this continuously growing. And of course, this also including several customized cells that can carry out particular function that is needed for a specific type of proteins. And in addition, developed by Yodai from Japan, they purified individually all the translation machinery from E. coli and reconstitute them again into a test tube, which can also perform the translation expression. And they are designed for applications that which will require very strict background and want to reduce the background expression to a minimum level, and this would be, of course, the best choice. So next, why we are using cell-free? So why we bother? We have so many diversified cellular expression systems for recombinant protein production. Why we bother to have cell-free? First, the cell-free techniques was not initially designed for recombinant protein production, rather than to study the machinery of translation, also a transcription translation coupled reactions. So it was not designed for recombinant protein production, but as a byproduct, we found that it can be very useful for a particular type of protein, which exhibit a toxic effect or a pressure on host cells. So this means cell-free techniques is removing the physical boundary of cells that does not have living cell and does not have cell membrane, cell walls. And of course, it's not a living system, so there is no limitation on cytotoxicity. So in addition, we do not rely on the intact cells. So in many cases, the chassis behavior of the cells, of host cells, does not play a big role. Of course, you can say there are still ingredients that are inherited from your cell lysate that can differ from one to the other. But in general, the core machinery as a central dogma would always play a big role or major role in our reactions, which can translate our the gene of interest to the target proteins. And of course, it first decoupling the growth and the catalyst. This is particularly useful for applications in metabolic engineering. So if we are interested in express or a set of enzymes that can fulfill the function of transform, for instance, from glucose to ethanol, and we are if we are using cellular system, we are relying on the accumulation of biomass first, which means we have to let the cell growing to certain or continuous growing until we can see the production of corresponding enzymes, and therefore the following catalytic reactions can happen. But in cell-free system, this does not have to be a must. So, which means in a cellular system, we would focus on optimizing the host cells, which would suit for a particular type of proteins or a particular designed metabolic networks, so that it can convert from one substrate to a final product. Or the cell-free system is simply crack the cells and reserve the most important part or most relevant part for central dogma, and we reconstituted the whole system as we want. So it is a fast and flexible system and platforms which can allow the protein expression within 2 hours, or we can extend the reaction up to here I noted as 12 to 16 hours. This can be easily extended to several weeks, even if you use controlled and continuous bioreactors, where you continue supply the substrate and you can extract your products, and this can really last for a long time. And of course, it is a flexible platform, and we get rid of the living cells. You can treat them as a simple biochemical reactions, and this is very easily be automated, which means you can really perform high throughput experiment using the automated liquid handling system. And this is the first prototype that I participated in back to my PhD time in Frankfurt, that we built a liquid handling platform, which we automated the whole process of cell-free protein expression, in particular designed for parallel protein expression tests and also condition tests, which we optimize the co-translational environment of our cell-free reactions, and especially for those are difficult to express. And lastly, as we said, the most advantage of cell-free system is that it's an open system. You can add any additives or chaperones, or chemical chaperones, or protein chaperones directly to the reaction tubes, so that your target protein can access such a beneficial environment as they were translated from the ribosome. As for membrane proteins, this is particularly important. As we know, membrane proteins needs the hydrophobic environment of cell membranes to be correctly folded, or in a more complicated cases, they would need a particular type of lipids or sometimes cholesterol and so on to shape their folding. And this is particularly used for when you use cell-free techniques, so that you can play around or tune in the hydrophobic environment, both from detergent or lipids, and the protein or detergent lipid mixtures, and so on and so forth. I'm gonna explain this later. And in another, I would not spend too much time on this topic. Cell-free techniques can also be used for fast prototype of metabolic networks for very complicated metabolic pathways. You can use different approaches to fast debug the whole network and see which is the rate-limited reactions of your metabolic pathways. And this can be very easily done by either mix the individual enzymes or express them in the same test tube in parallel. And this work is led by Michael Javert from Northwest Universities, or now he's moved to Stanford. So another, I would say, a big field of applications is in the past, I think in the past 10 years, is use cell-free techniques to reconstitute or assembly artificial cells. And this is particularly interesting is because we are now at the stage where techniques will allow us to see what kind of fundamental principles that the most simplest cells would need, for instance, to survive or to divide or to grow or to send signals, and so on and so forth. Of course, previously, the field of artificial cells developed from research communities that are starting from very simple materials like inorganic compounds and stepwise to transform it into complex molecules like amino acid or nucleotide. But now we're really moving into the stage that can we assembling cell mimicry systems using very simple molecules or even non-living components. And of course, cell-free technique is very useful. As I said, you can really fine-tuning or define your environment that can still preserve the basic transcription and translation function, which would give you opportunities to really synchronize all the different activities that exist or continues happening in a real cell. Okay, I'm not going to spend too much time also on this topic. So, and another very exciting application or combination is the combination of microfabrication or microfluidic chips and to cell-free techniques. This would allow us to really minimize the reaction volume of an individual reaction, really increasing the throughput several order of magnitude, so we can really running parallel expression like to the 10 power of 14, which would require which is a realistic numbers that if you really want to do let's say a task like directed evolution. And of course, this the cell-free system can be encapsulated into a micro, let's say micro compartment, like a drop water in all droplets or double emulsion vesicles. This represented a monolayer compartment, or like cell mimics like double emulsion biolayer lipid vesicles. And this, of course, can be achieved by this simple junction of PDMS chips, where you can observe expression of individual droplets on the high-speed microscope. Of course. Okay, let's get back to the topic. If we want to set up a cell-free protein expression system, what is the requirement? What do you need? So, as I said, we take the most well-studied or simplest cell-free system from based on E. coli cell lysate. It was not different from what you would need to express a recombinant protein in E. coli. You simply need a plasmid vector, which contains the promoter sequence and terminator sequence and the gene of your interest. However, in cell-free systems, it differs a little in the sense that you can also use a linear PCR template to express your protein as well. So as long as the promoter sequence and ribosome binding site is present upstream of your gene of interest, and this would be the only essential part that you need for expression in cell-free systems, which means you can really running a large number of expression reactions in parallel. For instance, if you want to study a mutation effect, or if we want to create a library of mutations for a particular protein, this is really helpful that you can skip totally the cloning step. And of course, you can move to the next round when you already get less candidates for beneficial mutations and so on and so forth. And also, if you have a large number of protein need to be expressed, they are totally different, and this is also save your time for cloning. And this can be achieved simply by standard laboratory techniques like two-round PCR that you could, including your promoter by first round of PCR, and then and of course the gene of interest, and then you amplify again the whole cassette of your PCR template. And of course, this is an example how we would vary the 5' sequence of a target protein that would not express with their native sequences. So we select several different tags where we intentionally tuning the composition of upstream sequence, where we see the effect of 5' upstream sequences. The only the reason why there are let's say variations in expression yield with different targets is simply let's say mainly because if the 5' sequence of your target gene, whether they would form a very stable secondary structure which shields your ribosome binding site, which would result in inefficient translation initiation. And this is, we found, the most relevant factors that would call leading to a poor recombinant protein expression. And okay, besides the template, what we need, as I said, so we need the transcription and translation machinery. So there are several for transcription, and cell-free systems always often use T7 RNA polymerase, which is really processive and also highly efficient. And there is also other polymerases that are used, for instance, the E. coli endogenous RNA polymerase, where this can be used if you want to tune or use the different promoter sequences to tune your expression. And of course, there are sp6 RNA polymerase, which is very frequently used in wheat germ cell-free systems. And of course, there are several other minor systems which are often used for particular applications. And the most important enzyme pores that fulfill the function of translation is coming from the cell lysate, which we crack the cells and followed by several standard procedures to fractionate the cell lysate and to keep the ribosomes, and as well as amino acid tRNA synthesis and translation initiation factors, elongating factors, and so on and so forth. And these are all preserved in our cell lysate. And of course, we added the amino acid substrate and nucleotide substrate. And of course, the most important is the energy regeneration systems. And there are really many studies that are dedicated into improving the energy regeneration systems using different energy precursors. And the most well noted is phosphoenolpyruvate acetyl phosphate, and of course coartem phosphate, such as which all have a higher phosphate energy bond, which can regenerate ATP from a single reactions and coupled to a particular type of kinase. And of course, there are different additives that you can choose to include into the cell-free system. This is, of course, highly diverse, so depending on individual cases. Okay, after we get the first basics of how the cell-free system is set up and consists of, let's move directly to membrane protein production. So as I said, the most challenging part for producing membrane proteins is how to get them into the right environment. So membrane proteins are always associated or integrated into a cell membrane. And why it was so difficult if compared to cytosolic proteins is that you have to provide sufficient amount of hydrophobic environment for overproduction, and very frequently the membrane protein that you are interested cannot be accumulated to a certain extent, and even you correctly target them onto the cellular membrane or plasma membrane, because it was already first, it was already very crowded with native membrane proteins, or secondly, if the target membrane protein that you want to express is highly toxic or cannot be accumulated too much onto the cellular membrane and will cause problem. Okay, so back to cell-free systems. So there are several strategies that you can use or start if you want to tackle the problem of membrane proteins, and we often categorize them into several key steps. First, as we discussed already, so we have to see if the membrane protein can be efficiently produced. Therefore, we need to optimize the basic conditions, such as the magnesium concentration, or if the native sequence of your membrane protein or your target protein is not well expressed, and you have to still optimize their sequence. In particular, 5' sequences of your gene, so that it can be well expressed. If the expression was achieved in relatively acceptable level, and we move to the next step, how we can get the membrane protein in their soluble form. And this can be achieved by introducing different hydrophobic environments, such as a detergent or lipids. Lipids can be categorized into either biocels. This is achieved by mixing two different short chain lipids, long chain lipids, or the most well-studied model lipid system like liposomes, or the recent planetary lipid bilayer is the Nano Letters. And this was formed by a scaffold protein and squeezed with different lipids so that it's formed like a discular structures. And in the middle of your disc, there is your lipid bilayer. So this all can be introduced directly into cell-free systems. Okay, so this is an early work back to the times when I was still in Frankfurt. We systematically studied an integral membrane protein, which means it at least has two transmembrane segments. So we systematically evaluate the expression of E. coli integral membrane protein, which consists around 100 individual proteins. They are diversified in their transmembrane segments, and also the size, as well as the number, as well as the extracellular loops, and so on and so forth. So after 3 levels or 3 rounds of optimization, so we achieved around 83% successful rate, which means around 80% of the integral membrane protein we can obtain in their soluble form. And next, I'm gonna move to a specific case that I involved during the past years for investigate the function or structure of a membrane protein. And I would want to start with the simplest channel proteins, which is the aquaporin. Aquaporin is a water channel protein, which is highly specific for the transportation of water or bidirectionally along the osmotic gradient. And it's a highly specific, and it was highly controlled. And of course, the most challenging part for study this type of channel protein is how we can accumulate enough materials for a function study. And the function analysis of aquaporins requires homogeneous distributed or homogeneous protoliposome, which means first we have to achieve integration of aquaporin into artificial vesicles, and the size of this resulting reconstituted vesicles has to be homogenized, and it's very tedious and complicated procedures that you have to went through if you do not apply cell-free. And of course, there is also a report that aquaporin can be expressed either in most cases in yeast, and you can isolate the membrane fractions of the yeast and to further purify the recombinant aquaporin. But of course, you suffer from the endogenous protein contaminates, and you also have to rely on if you have to isolate the membrane fraction, you have to went through tedious ultracentrifugation steps and membrane solubilization and protein purification and so on and so forth. And along the years that we continuously optimize our processes so that up to now, we can directly express our aquaporin or what channel proteins in cell-free system co-translationally with liposomes. And by two steps of treatment, we can achieve homogeneous or successfully reconstituted protoliposomes for the functional assay around 24 hours. And this is a very exciting sample preparation procedures that we completely omitted the purification steps, that you have to first purify the protein within a detergent and then reconstitute it or destabilize your liposome and so on and so forth. And this is really a big improvement for sample preparations for study aquaporins. Okay, the next very important type of membrane protein that we spend effort is the G protein coupled receptors. And of course, why they are very hot target, because most of GPCRs are disease relevant, and they are a directly drug target. And of course, the function of a G protein coupled receptors is depending on lipid environment and also on the oligomer state of the receptors and so on and so forth. And we really spend a lot of time to establish a standard sample preparation protocol that are suited for ligand screening, which of course is the key task that we investigate for GPCRs, because we want to develop a drug that is, really can tuning the activation or inhibition of GPCRs. And of course, a recent publication from a group in Frankfurt, that they exactly use they further develop this the systems so that we can not only express the GPCRs inside a nanodisk, but also couple them into their trimeric G proteins. And of course, such coupled complexes can be directly transferred or fused onto a living cells, so that we can perform both cells-based assays as well as in vitro assays. And this is really a milestone of how cell-free techniques can tackle the challenges of GPCRs, in particular their both structure and functional investigations. And of course, the cell-free system is very flexible. Besides the integral membrane protein, it can also solve the problem of membrane-anchored protein. A membrane-anchored protein is a very dynamic type of protein that is shuttling from cell membrane or cellular membrane environment to cytoplasm, cytosolic environment, which is tuned by their hydrophobic moieties. And in this case, we investigated the CDC42, which is cell division protein, which is the central molecules for development of cellular polarity. And the tricky part is this type of protein is really highly regulated. It interacts with more than 20 different proteins in a cell, and constantly shuttling from the plasma membrane and also cytosols. And of course, it's a leading signal for establishing cell tubulin as well as a cytoskeleton network. And therefore, to get sufficient and stable CDC42 is very challenging. But as I said before, the cell-free technique is or reactions are very flexible. You can simply deposit such a system in any environment that you want to investigate. So we directly put the cell-free on top of a supported lipid bilayer so that we can trigger the membrane targeting or the membrane extraction process directly on top of a supported lipid bilayer. And the whole processes, of course, can be easily monitored by a confocal laser scanning microscope. And therefore, we completely avoided the extremely or maybe mission impossible task to get first the target protein purified or later on the next step investigate how they are regulated or how they shuttled from cytosolic environment and to the membrane. And of course, there are recent following works. Another group from Japan is also implemented the same approach to address another lipidation proteins which happen in the N-terminals of a protein. And this really opens up how cell-free can address this type of highly dynamic or highly regulated protein that is associated or targeted or anchoring to a membrane. And of course, in summary, this is a short list of what we have done in the past years to achieve successfully via cell-free systems a list of membrane proteins that we investigated. This range from channel proteins, water channel, ion channel, and to complexes such as ATP synthesis, and also endotoxins like hemolyzing, and of course G protein coupled receptors. And with this, I would like to thank mainly to my former colleagues in Germany, and particularly from Max Planck Institute of Biochemistry and Goethe University from Frankfurt. And of course, I would like to acknowledge the help for all the fundings that allows us to continuously develop the cell-free techniques. Of course, thank you for your attention.
So thank you, Dr. Kai, for the enlightening lecture. So GenCefe Biotech, it was established in Wuxi, and we started in the US branch in California. So we are the company to development, large-scale production and application of the DNA, RNA, and gene synthesis. So we providing the CRO service, molecular biological reagent, and the pharmaceutical raw materials. Here is our platform. We have 3 platforms. We are working for the oligonucleotide synthesis and the modification platform, gene synthesis platform, and the safer protein expression platforms. For the oligonucleotide side, we are working for the raw material for the qPCR, POPs, and the primer, and NGS primer and POPs. For the researchers, we are working for the siRNA, microRNA, sgRNA, and the ASO. And of course, we are working for the packaging virus as well. For the gene synthesis platform, we are working for the gene synthesis and the content optimization. And if you have a request on the plasmid, we could perform the plasmid preparation. So further, if you find our service interesting and wish to learn more, please do not hesitate to contact us. We will ensure your inquiry could be reached and the prepared sales representative or distributors. So thank you for everyone to join our webinar. We are very appreciate your participation and looking forward to see you next webinar. Thank you very much.